So good afternoon, everyone. Welcome back to the post-lunch session of the FDP. And today we have, hello. Yeah. Today we have Dr. Ravi Krovidi, who is the CEO of Ingenuity Biosciences with us to deliver a talk. As a brief introduction, he is currently heading the Ingenuity Biosciences, where his function is techno-commercial business development of large molecules and clinical trials. He has over more than 18 years of total experience in industry and academia. He has completed his PhD in biological mass spectrometry from Max Planck Institute, Germany, and he worked as scientist at Pacific Northwest Labs, Washington, USA, and he has head Center for Advanced Protein Studies, Syngene International Limited, Bangalore. Earlier, he has received a BIRAC grant for establishing national laboratory under prestigious National Biopharma Mission program. And he was senior management. He had senior management positions at Lambda Therapeutics Limited and other multinational companies. He has worked for product-based biotech companies, and he was an invited speaker at various leading national and international conferences. His specialized area of expertise include drug delivery, discovery, and development, and contributed towards submission of CMC filings for various monoclonal antibodies. With this brief background, I invite Dr. Ravi Krovidi for his talk. Over to you, Dr. Ravi.
Yeah, sure. Thank you so much, Dr. Sundaram, for the introduction and for having me with the NIPER group members. So it's in fact indeed a great pleasure for me to share my experiences, my learnings in the field of biopharma, especially biologics, a little more focus on the clinical development side. So just before I begin, I would like to convey my sincere thanks to all the NIPER team members who have been very actively engaged right from the early beginning of the course designing. So I think it is a great opportunity for people like me from the industry side to share our experiences, and I believe all the participants, the students, the staff members, it will be a great learning experience. So I will continue my talk. It will go up to another 45 minutes to 60 minutes, as against 90 minutes. And you may pause me if you have any questions as I'm speaking, or I will be. I'm glad to take the questions at the end of the session as well. So whichever works best, I would leave to Dr. Somasundaram to handle it accordingly. Yeah. So if you may allow me to share my screen, I can present my slide deck.
Yeah, you can share right away. Okay.
So let me know once you're able to see my screen.
Yes, we can see it.
Yeah. So once again, good afternoon to all. As mentioned by Dr. Somasundaram, I head Ingenuity Biosciences. Ingenuity Biosciences is a contract research organization, which is a newly established. So I take care of this facility, and we have a team of, so it's actually established with VIDA Clinical Research. VIDA is a pioneer in the field of clinical research since more than a decade. And so overall, we have about close to 300 people working for large molecules and small molecules together. So Ingenuity Biosciences focus is mainly on biosimilars and biologics. So I'll walk you through, what are the developmental stages with respect to biosimilars, right from the early development stage to clinical evaluation. So I have structured my talk in two different presentations. So the first one will talk about, more about Ingenuity, so that, people may get an insight into what companies like Ingenuity Biosciences. I would focus on Ingenuity, but that would give you an insight into how products get developed by collaborating or by outsourcing to CRO laboratories. And there will be some case studies in my talk, and there will be some specific technology overview. For example, in terms of characterization, I believe my earlier speaker, Dr. Ravi Shankara, has touched this topic in great depth. But however, I'll try to give you a few more glimpses into the characterization side as what are all needed for advancing the product development from the early stage to the late stages of development, and how these attributes, the critical attributes, play an important role in the clinical domain. And then I have a little more focused talk on the PK and the ELISA, which is the pharmacokinetics, which is the ELISA-based techniques. So I'll be covering these two areas. So here are a quick overview of my talk today, starting with the biosimilars overview, and then the biosimilars development from bench to bedside. I have highlighted the key technological advancements. So this is something what CROs like Ingenuity Biosciences have introduced newly into the service offerings, which actually help in developing the assays at a much faster pace, giving the advantage in terms of the timelines and also the validated methods what we have. And then, of course, since students who are pursuing the master's program in NIPER would be keen to understand what are the requirements in case if you are considering opportunities to get into non-research based job profile, then I would say CROs currently there is no research, at least in Indian CROs, there is no research. Let me be very honest and upfront. But if you are looking for a job profile where you can actually, there would be a learning curve, there would be a learning opportunity, immense learning opportunity, and you would be working in a fast track mode as against a product-based company. So their project management becomes a very critical aspect, because some of the students, even PhD scientists who have finished, they would like to digress to a completely different domain into management. So which is not just the general management, but when a project is awarded, coordinating with the scientists, coordinating with the technical people, coordinating with the analysts, coordinating with business development, coordinating with clients, coordinating with regulatory requirements. So all this project management is a very strong portfolio. There are dedicated project management courses, but however, there are people who actually learn over the job. So this is another great opportunity where a lot of youngsters have been considering and exploring this option. So I thought I'll give you a little insight into project management. And in terms of the CRO requirements, for example, let's say tomorrow, if you are getting into a product-based company, in order to save time and save capex, the companies may be outsourcing their technical requirements. So in case if you are a person who is liaising these kind of interactions between the client and the CRO, so it is good to be aware of what are the CRO requirements you would want to consider for outsourcing a specific scientific activity. So that is these two ones. So these two I've highlighted in red just because this is a little more non-scientific, especially the number 4, which is more on the management side. And the third one is highlighted because this is something a differentiator. It's always good to have, especially in a CRO industry, a me too kind of an offerings will not help. It has to be a niche area, a scientific advancement, either in terms of the technology or in terms of the expertise and the overall experience. These are the two strengths which drive the business. So hence, these two are a little more deeper emphasis I've given. And then in terms of the scientific aspect, PK studies, which is basically on the ELISA overview, ELISA-based platform. And then I'll walk you through method optimization and validation studies of a specific PK assay. And then that's on the PEG-GCSF. Similarly, on the study samples and so on and so forth. I may pause at point 10 at the immunogenicity testing in considering the time limitation. So we'll see how much we can cover in today's talk. I hope this is acceptable to you all. If you have any questions, I can pause for a while and I can take those questions. For some reason, I can't see the screen of all the members. I don't know, is something wrong with my thing or, yeah.
No, while presenting, I think you cannot see others.
Okay. Then I may not be able to see unless they verbally express. I can't
Yes.
see if anybody is chatting. So then I'll
Yes.
continue with, I can just see only my screen. So I'll just continue with that.
Yes.
Okay, sure. Yeah, so this is, if you look at the biosimilars overview, so biologics are effective. These are life-altering therapies, which are certainly expensive. And there's a limited access because of their expensive price tag that is associated. And the good news is there are many originators or many innovator molecules which are actually nearing their end of the patent life. So once their patent life is getting to an end, it gives an opportunity for another company to get into that product development and quickly develop the product and market it. So there's certainly a high need for these quality biological therapies. One is affordability and then accessibility. These two are the critical aspects for developing any biological therapy. That is, as I mentioned to you, the innovator molecules are highly expensive. So once the patents are off, it becomes, of course, these are complex, challenging molecules, but the goal is to make them affordable, and the goal is to make these biological drugs more accessible. So if you look at a technical definition of what's a biosimilar, a biosimilar or a biological molecule is a biological medicine where you do not see any meaningful, does not have any meaningful differences from the reference medicine in terms of the three critical aspects: quality, safety, or efficacy. That means the two molecules, the innovator and the biosimilar, essentially should not compromise on the quality, safety, or efficacy. So that means there can be subtle differences, but those should not be any having any implications. So they should not be having any meaningful differences. Or we can elaborate a little more, saying that no clinically meaningful differences. So when you say clinically meaningful differences, whether a patient selects an innovator molecule which is more expensive, or against the biosimilar version of it, which is an equivalent version of it, it should not have any meaningful difference, a clinically meaningful difference in terms of the safety of the molecule, the purity of the molecule, and the potency of the specific molecule. So all these three critical attributes need to be, these are sacrosanct. They need to be, ensure that these three are very well maintained in order to qualify them through the regulatory agencies. Now, coming to these, there are a few little scientific jargon which is good to be aware of. For example, when you look at a biological product on the third column, you can see definition. Any virus, any therapeutic serum, toxin, antitoxin, vaccine, so on and so forth, or all these, they fall under this huge basket of biological product. And an originator, sometimes, there are different synonyms. Some companies call it as a reference, some are innovator, some are comparator. There are different geographical regions, but they all refer to the same thing, which is an originator or an innovator molecule, or the molecule which has been first developed or designed and developed in a particular laboratory. So, and of course, since there is a lot of investment in terms of time and effort, and a huge amount of resources have gone into it, so these molecules are highly patented. When I say patented, the molecule, it need not be just the structure of it. It could be the process associated with it. It could be the manufacturing thing that is associated with it. For example, adalimumab has highest number of patents, and it makes it very tricky and challenging for a biosimilar company or a new company to develop a biosimilar version of this molecule, of this originator molecule. In terms of the biosimilar, you can see they are called in different geographies, like follow-on biologic in the US, in subsequent entry biologic in Canada, biocompatible in Mexico, etc. So now I would not go more into the definition of biosimilar. By now, I believe you're all familiar with the word biosimilar, which is a mirror copy of it, having no clinically meaningful differences. There's another version of it, which is called biobetter, which is a second generation biologic, which is biosuperior. This is also based on an originator medicine, but with improvements to increase the efficacy, potency, marketability, safety, etc. So it's a better version over the biosimilar. The copy biologic is an alternative biologic, and the copy of an originator medicine that has been approved in a country where no official biosimilar regulatory pathway existed or has been existing, so on. So that's called as a copy biologic. So these are the different terminologies, but let's kind of focus more on the biosimilar in the next few slides. And if you see, these are all the different commercial biologics that are available. So if you see on the left side, starting with the erythropoietin EPO, GCSF, HGH, IFN alpha, IFN beta, and then you can see their commercial names: Ipogen, Neupogen, Genotropin, etc. And then you also see their branded names. So and on the right side, you see the MABs, monoclonal antibodies, which are complex biological molecules having the light chains and the heavy chains. So starting with the etanercept, adalimumab, bevacizumab, Cetuxy, Inflixi, so on, and then the trastuzumab with their branded names as Enbrel, Humira, Avastin, Erbitux, etc. So these are the well-known. If you are interested or if you are pursuing to get into a pharmaceutical industry in India, predominantly these are the MABs which have been primary focus for the Indian biopharma industry to start working on these biosimilars. So the tier 1 companies, if you see in India, most of them are actually heavily focusing on these molecules. So it would be good if your interest is to get into biologics or especially into biosimilars, to understand a little more on the what it takes to develop this right from the clone stage to the clinic stage. From the clone, from the characterization of the clone, from scaling up of it, right from shake flasks, scaling it up, purification, characterization, and then the clinical studies, or initially the preclinical studies follow the clinical studies. So it would be good to have a deep overview into those. Now here, if you look at the difference between a small molecule and a biological medicine, like generics, as you all may know, India is considered to be the generics capital of the world, because we have been pioneers in the field of generics, especially in small molecules domain. So the vision a lot of companies have is to achieve the same level of reputation for biologics, especially biosimilars, where we can actually bring in new technologies to make these drugs more affordable and accessible. So there are different manufacturing processes that can and often do yield differences in the end product. As I mentioned to you, the product may have differences, but it should not be having any meaningful clinical differences. That is what will qualify a specific biosimilar. And after the quality of a biological medicine is demonstrated, some non-clinical and clinical studies are necessary. So that is, preclinical studies and clinical studies are necessary for ensuring that the biosimilars will be launched into the market. And then the immunogenic responses, that is the acceptance of any foreign biological material by the human body, that is gives the immunogenic profile. So the immunogenic response cannot be predicted, so therefore it must be tested. And all these battery of tests, especially there are about a battery of 12 to 15 tests that need to be performed to ensure that the product is maintained with highest quality. There are different biophysical characterizations which are called as advanced analytical characterization techniques. And then there are functional characterization studies that need to be performed, especially the binding studies, to make sure that the monoclonal antibody is indeed binding to its specific receptor on the cell. So there are techniques like is the surface plasmon resonance or a mesoscale discovery. These, I'm not sure how much of these have actually trickled down into the academic level lectures or academic textbooks, but if you actually read scientific publications where if you ask a question, what are the binding targets of a particular map, what cell lines are used to evaluate the binding efficiency of this, what technologies are used in, for example, if you look at the Amgen's molecules, there are n number of publications you can see in terms of the Biacore based specific binding interactions. So all these tests have to be performed. There's no AI-based prediction tool to give you an upfront indication of how the molecules might perform. So that is not possible in this specific domain. So this is a quick recap of the earlier slide. Again, if you look at from the timelines from 2010 to 2015, there are about 99 molecules which have gone off patent. And then 2016 to 2020, and then you can see the number of molecules that are losing off patent in terms of giving an opportunity for these molecules to be developed. And here again, we know what should be compared with the reference product. So in vitro studies generally need to be performed to assess any possible differences in bioactivity. That is, binding to the target antigen. As I mentioned to you, the cells have the cell surface receptors, which are by a lock and key mechanism, the antibodies, the monoclonal antibodies bind to the specific cell surface receptor, and then as a cascade of signaling mechanisms happen, finally leading to the turning off the cell in case if it is a proliferation. For example, if it is related to a tumor-associated specific biosimilar, then the replicative efficiency of the cell is turned off once the binding is achieved between the monoclonal antibody and the cell surface receptors. So how do we assess these? Like, for example, the Fab associated functions. So if you look at the monoclonal antibody, it has two components, the Fab and the FC region. So the Fab region is the one which has example, neutralization, the receptor activation or receptor blockade is the critical part in the Y-shaped structure of it. The stem of it is the FC, and the two projecting arms on the left and the right are the Fab regions. So, and the FC associated functions, they have a very critical component in the ADCC and the CDC assays, especially in the complement activation. So I'll give you a little more insight into this kind of immunogenicity examples for the FC associated functions. And in terms of the safety, pharmacology, reproduction, mutagenicity, and carcinogenity, generally these are not required, which are otherwise required, and there's no extensive tox studies that are needed for biosimilars. So which means less animal sacrifice for a biosimilar development. And there are also a lot of voices coming from the industry trying to remove or trying to mitigate or trying to lessen the number of preclinical studies that are needed for a biosimilarity assessment. Because fundamentally, if you look at the human biology and the animal biology, to a certain extent they may, but by and large, there's no significant value coming from doing these in a preclinical setup. And then coming to the in vivo studies, it depends upon the need for additional information. As I just mentioned to you, that there are a lot of voices coming in the industry, and there are recommendations to make sure that people are more cognizant of what preclinical animal model needs to be selected. Is it really needed? Or like, for example, if the relevant animal model is a non-human primate, so the FDA EMA generally do not require an in vivo non-clinical study unless it is absolutely needed. So the regulatory agencies are very much supportive as far as there is a strong scientific and a rational behind the justification. So this is a quick overview of how the biosimilars nomenclature has been set in globally. Just like, for example, if you see in the IUPAC nomenclature, which is a well-known established nomenclature in the field of chemistry. Similarly, in the biosimilars, so if you look at the table, starting with the column 1 with the mAb, either it is a UMAB, a SHE-mAb, a ZU-mAb, or a MOMAB. So what is U when it is a completely if it's a human monoclonal antibody, it is adalimumab, where it is entirely derived from a human source. That means very less immunogenic related issues. If it is a chimeric mAb, so you would see XI, which is a rituximab, infliximab, cetuximab. So that's XI refers to that where the variable regions are coming from murine and the constant region are coming from the human region. So that means these are a combination coming from both murine and human, which definitely signals, or there's a red flag saying that there's a high possibility of immunogenicity, immunogenic responses. So one needs to do an extensive battery of immunogenicity studies. Similarly, if it is ZU, it is completely humanized. These are mostly from human except the binding region. So that is the binding region. In the Fab region, there is a CDR, which is the complementary determining region. So that is completely, mostly these are from human except the binding region. So for example, the trastuzumab and the bevacizumab. Similarly, Mo is like a pintumomab, bectumomab. These are entirely murine source. So certainly, since these are non-human, there is a high possibility for the high rate of immunogenic reactions. So immunogenicity is a very critical, anything which is non-human. And this is again another quick one where you can see the initial approval and the revised approval, and then the innovator in the third column. So just to give you a quick glimpse of when they got the initial approval, and how long they had the patents, and giving an opportunity for the other companies to start getting into it. For example, if you see the innovators in the third column, the Genentech, Amgen, Eli Lilly, these are all the companies who have actually invested a lot of time and effort in designing these monoclonal antibodies. They're developing these monoclonal antibodies and doing extensive clinical research, and then launched into the market. But unfortunately, we do not have any Indian companies there yet. But now there's a lot of focus that's happening in many Indian companies where novel molecules are being tested and evaluated for biological specific biological areas. Yeah, so this is a quick overview as what are the the development trajectory for an originator and a biosimilar. So on the top you see, for example, for an originator, the RMP sourcing and logistics. Then there is an amino acid verification that happens. Then there is a quality QTPP establishment. Then all the methods which are used for analytics they need to be qualified, and then these the quality target product profile has to be established using a battery of about 12 to 15 tests, which I just mentioned. As these methods get developed and they get transferred to a laboratory where there is extensive high throughput screening and a complete production of this happen in every single batch. And then there are stability studies, both short-term stability and a long-term stability. The shelf life and accelerated studies happen. And in the below, you see there's a clone selection, beginning with the clone selection. There's a media selection. There is a process development. And then the biosimilarity, comparability studies, etc., followed by method validation and release testing. So if you compare these two from the top and the bottom, so of course the in-between one is pretty much overlapping for both. It starts with the cell line development, there's a media optimization, there's upstream development, so on, and finally moving to the approval. But the bigger advantage of developing a biosimilar is, number one, it is low risk, because it's like getting into the woods with a map in your hand. So you know the directions, which way to go into the woods. So you are less lost, I would say, because you know what you are looking for. You know what molecule, what modifications may perhaps influence the immunogenicity. You look for, because you have the amino acid sequence in hand, so you need the right tool to make sure that your molecule has indeed the same amino acid sequence as what you started with no modification on it. For example, post-translation modifications, glycosylation. So you have the map in hand. So the responsibility of a scientific person is to make sure that this is replicated consistently, and replicated the yield is high. So there's no point in making several batches, but the yield is less. So it is not commercial. It will not be commercially successful. And then the other important thing is the testing of the host cell proteins, the host cell DNA, which are process-related impurities, because since we are handling with the CHO cell lines, there is a high possibility for this host contamination to be coming in. So there are specific techniques to ensure that there's a host cell protein and host cell DNA are present in a very, very minimal under regulatory specifications. Yeah, this is a quick, in terms of what Ingenuity or recently VIDA Clinical Research has also acquired BioNeeds, which is completely into preclinical domain. So we offer even preclinical testings, starting with in vitro cell-based assays, as I mentioned to you, Biacode, mesoscale discovery, or octet-based studies. So these are the ones which go on. And then from the animal side is the in vitro immunogenicity. It could also be cascade immunization. For example, there are commercial kits that are available for a particular product. But if this product has to be, if the antibodies have to be raised against the specific in-house monoclonal antibody, then they will not be commercially available. So what one does here is take the innovator molecule, and then they inject into the animals the polyclonal, they put it into goat, rabbit, and then they raise the polyclonal antibodies over a cascade immunization process, purify it, and then they do a titration. So, and then do the immunogenicity studies. And similarly, preclinical toxicology studies are needed. And I touched upon the host cell proteins and the host cell DNA. So these are all, a gamut of tests that need to be performed once the product is actually out from the initial purification step. And similarly, before that, a lot of work has to be done in the cell banking and cell line characterization. For example, the preparation of the master cell banks, preparation of the working cell bank, the viability analysis, and the basic cell line characterization. For example, it could be contaminated with mycoplasmas, and then look for the doubling time, look for the viability of these cells, etc. So all these have to be extensively done in a very, very meticulous manner to ensure that there are no hurdles in the initial stages. Similarly, other preclinical services, there are bioidentity, biopotency, TK studies, and then polyclonal I just mentioned to you. And similarly, for the impurity, the host cell protein and the host cell DNA. Similarly, the purification of the anti-serum using protein A and affinity chromatography systems just done on ACTA. And then the critical reagent preparation and labeling. For example, if you're looking for reagents that are needed for ELISA, so those reagents have to be of the highest order. And there are commercial kits that are available, but this is another area in case as a scientist, if you would like to get into that. These are purely technical, which are a lot of biochemistry is involved, a lot of molecular biology is involved in getting into those domains. And in terms of the key technological advancements, what Ingenuity currently has is the MABY, which is a recombinant antibody platform. Then there's a powerful software, which is a lab informatics solution called as the AGRIS. And then there's a biosimilar characterization solution called as an IntelliB. So these are the three innovative technologies. So if you get a chance, look into the there are other many such innovative technologies that are available. So here comes an opportunity for students to actually think of how new solutions can help in advancements of the product development at a faster pace. For example, if you see lab informatics solutions, the lab informatics solution provides a comprehensive solution, a service offering right from the raw materials that are used in the product in the early R&D stage, then in the production stage, in the testing stage, and also the statistical. AGRIS is a very powerful statistical embedded tool which is used for PK ELISA based studies and immunogenicity studies. So people who have statistical interest can develop algorithms for which can be deployed for doing the cutoff value determination. For example, when people are working on immunogenicity, there's a high level of background signal to noise that comes in. So the cutoff is what makes a huge difference. When I say cutoff makes a huge difference, it is separating the signal from the noise. If you are not able to separate the signal from the noise, then there could be a lot of misrepresentation that might happen. And the way to filter that is there are commercial softwares available, but developing a software using a wealth of information which is published and then developing a statistically based strong software makes life easy for the scientist as also for the data that gets generated. And then the other offering what we have is there's about 120 validated methods, especially for with primarily 70 biosimilar molecules. So when I say validated methods, it means that these methods are readily available. Generally, the journey that begins in biosimilar industry is developing a method, qualifying a method, validating a method, and then doing the sample analysis. It's like, in the space station, the rocket being lifted off, or at the airport, where the flight journey probably is only, from Kolkata to Bangalore, perhaps it's about 2 hours or so. But it's not actually the journey time. It's actually the preparation time which makes it a lot of difference. But if everything is all set, it goes in autopilot mode. Similarly, if you have a validated method, it can be considered that the entire assay will be performing in a seamlessly smooth autopilot mode. And either it is by the experience of the analyst, or it can also be made in a robotic fashion using a specific robots. So that's the advantage what we have when we say any laboratory, for that sake, not just anyone having a validated method means that the method is robust. The method has been specifically tested for this molecule, and the equipment which is used is indeed very specific. They do the specificity runs to make sure that the method is suitable for the molecule, etc. So these are it needs to be qualified, it needs to be validated. And in terms of the regulatory experience, there are three major regulatory agencies, I would say, globally. Of course, there are many, but the three ones, the largest, as you know, US is the FDA, and then the European EMA, and then the Brazil one. So these are the three major. US markets is still one of the largest markets in the world, and the companies, and then comes the European market as well. So the companies which are focused in developing products for launching into international markets, then they need to fulfill the conditions, the specific requirements as per the FDA and the EMA guidance. I'll skip this. This is a quick overview about, again, R&D, preclinical to phase 1. This is what is a one-stop solution, what we offer in VIDA and Ingenuity Biosciences, where there is a R&D associated, especially with the softwares which I just mentioned. There's a preclinical wing, which is in BioNeeds in Bangalore. And then we do the phase 1 clinical trials in VIDA Clinical CRO. Yeah. And then the three key technological advancements, to go a little deeper into that. One is a biocompatibility testing solutions. So we offer a wide range of biocompatibility testing solutions so that the biosimilar molecule can be tested in the early development stage. So there's a minimal risk of failure, etc. And then the solutions that are needed, in terms of the bioassay solutions as such, that is the entire battery of bioassay solutions, the cell-based assays, which are used for understanding the drug mechanism of action. Like, for example, the cell proliferation, the cell viability, cell signaling, receptor activation, LBA, all these which are used to measure various downstream proteins. So there is entire bioassay services using ELISA-based, using Biocor-based, using mesoscale discovery technology-based. So these are the offerings what we are currently engaged in. Similarly, the antibody functionality testing, which I mentioned, like, for example, the FAB region and the FC region. Especially in the FC region, the ADCC assays, the CDC assays, where there is a FC receptor binding assays. So if you look at the stock of the monoclonal antibody, the FC gamma receptors, the FcRn receptors, these are the ones which determine the half-life of your monoclonal antibody. So these play an important role, and testing these is a critical aspect. So this is a quick overview of the different MABs that are currently, as I mentioned to you, we have validated methods. So this slide is a very intense slide, which takes about close to 1 year to develop these values. So starting with the product name, you can see in all the columns, in the column starting with cetuximab, rituximab, you see all the values from top to bottom. For example, what is the detection method? What is the precision CV that was achieved? What is the accuracy? What is the sensitivity? And then the dilution linearity and the hook effect. So you see all these technical parameters, they will be evaluated and rigorously tested. And finally, the assay is considered to be a validated assay. Yeah. So I will skip to the next part. Yeah. So this is I think we have yeah. If there are any questions, I'll take it. I'll pause for a while. Okay, I will continue now. So this is an ELISA overview PK studies. As you all may know, ELISA is also known as an enzyme immunoassay, a biochemical technique which is used to detect the presence of an antibody or antigen in a sample. It's typically performed in a 96 in a high throughput manner, or it can be done in a 384 also. There is essentially what you have is a two components, is a plate reader and a plate washer. And here, binding and immobilization of the reagents happens. So it makes ELISA easy to design and perform because you know what is binding, what is immobilized. Either your receptor or your antibodies is immobilized, and the other one gets bound to the immobilized component. And it is easy to separate bound from non-bound materials by different wash conditions. And this is a powerful tool for measuring specific analytes within a crude preparation. For example, from serum, if you would like to see what components of your serum are bound to the immobilized component, so you don't need to do any extensive sample preparation to understand the specific interactions that are happening between the bound analyte and its target present in the complex biological fluid like serum. So ELISA has been used as a diagnostic, which is both qualitative and quantitative tool in medicine, in peptides, in proteins, antibodies, and hormones. So ELISA technique broadly can be considered into four different ways: a direct ELISA, a indirect ELISA, which is a sandwich ELISA. This is also called direct ELISA, sandwich ELISA, and then the competitive ELISA. So if you look at the sandwich ELISA, the coating of the plate surface with known quantity of capture antibody. So your capture antibody is what goes it's coated onto the plate. Then you block any non-specific binding sites on the surface. Then you add the antigen containing sample to the plate, which is coming from the biological fluid. Then you wash it to make sure that anything unbound is just washed out. And then you add an enzyme-linked detection antibodies, which also bind specifically to the antigen. Then the washing of the plates, again, to remove unbound antibody enzyme conjugates. And then the addition of the chemical or the enzyme substrate, which is converted by an enzyme to a colored product, which is eventually read out. So that is, you measure the absorbance and you determine the presence, and then estimate the quantity of the antigen that is present. So that is the sandwich ELISA. So initially what needs to be done is you need to do a method optimization or a familiarization. So for any biosimilar product, so there is whole wealth of information which is available. So there are a lot of desktop research has to be done initially to understand what has been published, what are the values, and in terms of the clinical pharmacology section, which determines the dosage, the time period of dosing, etc. And then a clinical study samples may have analyte, which is a biosimilar or a reference concentration in wide range, depending on the time points and its pharmacokinetics. And then once you have you thoroughly evaluate, review the literature, so you arrive at the Cmax, the half-life of the particular monoclonal antibodies. You basically look at that, and once you have the Cmax data, then you design your PK assay. So then you need your Cmax, as I mentioned, is a half-life. So you have the quantification curve determined in such a way that it fits into the entire linearity part, the ULOQ, LLOQ. Then once the ELISA reagents are received, so you procure the reagents, ELISA reagents, which are commercial kits these days. So you procure them, you stock them, you store them in cold. And then before proceeding for your experiment, what you need to do is you analyte, all the analyte stock solutions are prepared in appropriate solvent, and the biosimilar and reference formulations are used as standard compounds to prepare the stock solutions. From the stock solutions, you make your spiking solutions, which are your working dilutions. So you prepare these That it is just like, as I mentioned to you, making all the reagents and ready before the assay is performed. And these are very critical, by the way. Yeah. And then you also have the calibration curve standards, which are prepared, which are generally 8 to 10 standards from the spiking solutions. And then you have the anchor calibrators, which are also prepared based on the method specific requirements. Then you have the ULOQ, LLOQ, the upper and the lowest limit. These calibration curves are selected such that it will be approximately 2 times of the anticipated Cmax of the formulation in the clinical study. As I mentioned to you, from desktop research, you look at the Cmax, then you design the curve, with the ULOQ and the LLOQ, especially approximately 2 times of the Cmax, and then your LLOQ should be approximately 5 times of the Cmax or lower. So there should be 2 separate sets of QC controls to make sure that there are no errors in your assay, and the QC should be performed from both test and reference stock solutions. That means your biosimilar and your innovator. So there are totally 5 QC levels as shown: ULOQ QC equal to ULOQ concentration, HQC, which is about 75% of ULOQ, and then the middle QC, etc. So these are all very stringent protocols, which are all drafted before the experiments are performed. And then the CCs and the QCs are prepared by spiking the spiking solutions into the blank human serum or plasma. Your spiking volume should not be more than 5%. So these are all the specific conditions for making sure that your assays are being rigorously. So the persons who are delegated, and they need to be trained. And so there's an extensive training program that happens before one gets into the bench for the validation activity, or even for qualification, I would say. And these trained persons carry out the method validation as per the method validation protocol. So a method validation protocol, as I mentioned to you, it's like having a map in your hand, a Google map in your hand when you're going into a new city or a new location. So you have everything very well structured, like point A, point A1, point A2, take a right, take left, so on and so forth. So your validation protocol actually has all these details very elegantly, very meticulously captured in a step-by-step manner. And then the analyst who is trained will perform the assay, and of course, there are SOPs which are written very clearly, like what pipettes to be used, what are the storage conditions, what are the the thaw conditions, the benchtop thawing, what are the specific conditions for sonication or for centrifugation, etc. There are very clearly drafted SOPs at the heart of the experimental activity. Anything written in the SOP is a religious, it has to be followed in a religious manner, step by step, with no deviations. If there are deviations, one has to mention why they had to deviate from what is already written. So this is a written guidance which is helping the analyst to perform, so that there is a high level of reproducibility whether it is analyst 1 or analyst 2 performing these experiments. The method validation experiments are done as per current requirements. This is what every CRO does. So there's no rocket science here. It's a very written, structured document, and they follow it. And all the samples are analyzed in duplicate, and they mean OD values are used for further calculations. So the results of all experiments are calculated using rigorous software, which I mentioned to you. Once you have all the data coming out from your experiments, they apply a software. Intense statistical capacity, and then you actually take the mean, and then you compute the values in terms of what is the binding efficiency, etc. So if you look at the experiments, so these are the experiments what are generally performed. The selectivity in 10 normal human normal lots, this is a normal serum, one each of the hemolytic and the lipemic lot. Accuracy and precision, we do 6 batches. Ruggedness, sensitivity, specificity, dilution integrity, concomitant medication, that is if somebody is taking a parallel medication. So what is the influence of that? And then the stability, the solution stability, as well as the matrix stability, like the benchtop, the freeze thaw, the long-term storage and freezer, etc. So all these are a part of the experiments that need to be performed during method validation. And all the method validation experiments results are compiled on a day-to-day basis. So using the Alcoa principles, which is the a norm, which is also very closely watched by the QA. And then there's a draft method, as I mentioned to you, there's a validation protocol, and then you perform your experiments as per the validation protocol, and then you capture the results, you run them through your statistical software, and then you prepare a validation report. So all data that is generated during validation along with the method validation are internally reviewed by a QC department and a QA department to make sure that everything is performed with no manipulations. And then after the completion of the QA review, the method validation report is signed off, and all the raw data are archived. This is all for regulatory. If you are doing for R&D, it is not required. Because you are still in the development stage. So there is a lot of flexibility. There's a lot of because in the development stage you are initially evaluating to get that your scientific opinion, scientific understanding of how the molecule behaves. So once you have the initial R&D has a lot more flexibility in terms of less paperwork, less stringency in designing the experiments, less quality. What do you call? Deviations that one may take in. So there's a lot of room to explore multiple parameters, multiple buffer conditions, multiple test run conditions, etc. Here is an example of a PK study using a PEG GCSF. So the PEG filgrastim, it's a 39 kilodalton, which is a pegylated form of filgrastim. GCSF, as you all know, it's a human granulocyte colony stimulating factor, and having a PEG associated with it, so it's a PEG GCSF. So the pegylation of the molecule, it's about 20 kilodalton, the polyethylene glycol. What it does is it extends the half-life of the protein for about 4 hours to around 15 to 18 hours. That means less number of doses for a person to consume. So it's sitting inside the bloodstream for a long number of hours. And so PEG filgrastim is widely used for stimulation of bone marrow to produce and increase the WBC count. And this is by far, high selling market drug. And the PEG GCSF assay employs the sandwich ELISA, which I just mentioned in the beginning as how the sandwich ELISA is performed. Then here we use a commercial kit. We have partnered with Somru, which is a Canadian company, and all the 42 methods that I've shown, the list of all the 122 validated methods, 70 for biologics, for biosimilars. So these are all established, these are all validated. So you have seen the extent of time and effort that goes in developing a validated assay. So these are commercially available, and there are different catalog numbers associated with each biological product. So here in this assay, the catalog SB037044 was used. And then this is a quick overview of the method. The antitherapeutic is coated onto the plate, as I mentioned, since it is ELISA based. And then the therapeutic region, which is present in the sample, which is your analyte, is bound by immobilized antibody. Then you wash it. Then following the wash, then you conjugate it using streptobidine reagent. And then then again you wash to make sure that the unbound step is removed. And then finally you have the color development, which is proportional to the quantity that is bound to it. So, and then you quench the reaction. And finally, you measure the intensity. So these are the results. So you can see, as I mentioned to you, there is a statistical software is used for the evaluation of it. And what you can see is all the values from the interbatch precision, the mean concentration in picogram per mil, the standard deviation, the percentage CV, all these get computed once you plug your values into the AGRIS software. And then we also check is there a hook effect or not. And once we have all this done, then the study sample analysis using the validated method. So what are study samples which are coming from the clinical side? So the study sample analysis is done once you have the method is validated. Then your bioanalytical study plan is prepared. So this is actually a everyday life, how the life of a CRO scientist is. So he would actually make a bioanalytical study plan before initiating the study. And then a team of persons is delegated and trained for performing this task. The analytical batch is processed in a single plate with one set of CC standards, at least 2 sets of QC standards containing HQC, MQC, and LQC. And then each samples are processed in duplicate so that there is no error and the mean OD is taken for further calculations. And then all the batches are processed as per the SOP, which I mentioned to you. And all calculations are done in using AGRIS software. And in case of there's a repeat analysis, samples from rejected batches are reassessed in repeat analysis. And the individual samples from accepted are also reassayed. In case of reassayed samples, only the repeat results are considered, etc. So, this is how the methods are run. And in case of incurred sample reanalysis, the ISR of already assayed and accepted samples in the study is done to check the reproducibility of the method. For that purpose, one sample at Cmax and one sample in the elimination region is taken from the randomly selected subject. So there are certain cutoff values as how the ISR has to be estimated. For example, per study, 10% samples are to be reassayed for ISR. In the case of samples are less than 100,000, if samples are more than 1,000, then 10% for the first 1,000 plus 5%. So there is a scientific statistical logic in considering the incurred sample reanalysis, the ISR. Then all the study analysis and results are compiled on a day-to-day basis. Then once you have the study completed, there's a draft study report which is prepared. All the data generated during the study analysis will be reviewed by QC and QA, because these are all going for regulatory submissions. And after completion of the QA review, the study report is further sent to the sponsor. And then after completion of the sponsor review, the study report is signed off and the study raw data are archived for long-term storage. So that is how overall, things on the journey of developing an assay in a clinical scenario. I'll slightly change my my discussion to a different one. Yeah, a little bit about VIDA. If you get an opportunity, you can also look into VIDA website, where it will tell you about what VIDA is. It's a global clinical development partner offering, it's the largest CRO in India. It has a robust quality compliance. It's a one-stop solution for complex studies. It's a skilled personnel with focus on continuous professional development, so on and so forth. This is a typical BD slide in case if you're, some of you may be interested in getting into a business development domain. So it is very important to ensure that the key capabilities are presented in a very effective manner. And so these are some of the capabilities what we have in our facility. There are about 46 LC-MS machines, which are mainly for small molecules. And then there are 2 ICP OES equipment, and it is all run on the Watson LIMS, which is the data processing one. And in terms of the storage capacity, there are about 45 refrigerators, which are where the samples have to be stored at -80 degrees. This is a new, when Somru got inaugurated, we actually appeared in the New York Weekly, where our joint partnership, because Ingenuity Biosciences is a joint venture between Veeda Clinical Research and Somru Biosciences, which is our counterpart in Canada. And in terms of the so these are the other equipment what we have. For example, flow cytometry. If you're looking for cell-based studies, the flow cytometry is essentially needed, and mass spec for doing high-end characterization studies, for example, intact mass, looking at the peptide mass fingerprinting studies, looking at the post-glycosylation modifications, looking at any N-terminal cleavages, etc. For that, the mass studies have to be performed. And then what you see on the bottom is the HPLC. Then there's a Orbitrap. And then the BSL-2 cabinets, where the scientists are performing cell-based studies, where they culture the cells and then they treat them with different conditions of, it could be stimulation or it could be proliferation, etc. And then the ELISA reader in the bottom right. I'll skip this. These are all, So this is one example of where studies have been performed on the mass spec using a trap with 10 HPLC. And then the software default comes with the software, the thermo Calibration finder software. As is, if you're looking as of it, or the sub of it, or you do a reduction like ATD reduction, you get the light chain heat change. So you can assess these critical attributes using such high niche technologies. Similarly, modifications, as I mentioned, glycosylation, or post-translational modifications like phosphorylation, etc. If you are more in omics studies, and peptide mapping and sequence coverage. 100% sequence coverage is what is critical to make sure that the biosimilar which is developed is indeed a mirror to the innovator molecule. Then the N-terminal and the C-terminal analysis. I mean, for N-terminal, a lot of, in the earlier days, people were using Edmond degradation. But of late, mass spectrometry has taken over Edmond degradation. But however, there's still some benefits of Edmond, but predominantly it is mass spec, which has replaced both for N-terminal and C-terminal in for protein characterization studies. And the other area is the disulfide bonding analysis, especially the disulfide linkages, which are present in the in your monoclonal antibodies. These are very, very critical. So doing a disulfide linkage analysis using HDX is very critical to ensure that the biosimilarity maps between the innovator and the biosimilar molecules are established. Yeah, I will. These are quick glimpses of how the profiles will look. So what you see on the left side is the size exclusion HPLC with the retention time of the innovator and the molecule. And at the bottom, you see in a reverse phase the trastuzumab, and there's an in-house clone that was developed. And on the right side, you see the peptide mass fingerprints of getting 100% sequence coverage in both the molecules, saying that these two are identical since their peptide mass fingerprints shows a high degree of overlap. And then at the bottom, you see the glycoforms, especially if you're there's an enzyme called PNGSF. If you use the PNGSF enzyme, the glycans get cleaved off, and then you can actually see the non-glycosylated version of it. But if you do not use it, a deglycosylated version can be will be produced once you use PNGSF. Yeah, this is another example of a ligand binding assay, which I was telling you, by ELISA-based techniques, where you have on the x-axis the concentrations in nanogram per mil, and then the absorbance, which is based on the sandwich ELISA. As I mentioned to you, there is a fluorophore, and then you actually get to see the binding efficiency. So this is another example of the intact mass analysis performed in the glycosylation stage, where you can see the adalimumab. Then you can see trastuzumab, denosumab, bevacizumab. All these molecules were not subjected to deglycosylation, and they were subjected to intact mass analysis. What you see is a deconvoluted mass spectra of 148 kilodaltons, 147, 148.06, and 149.19 for different maps. So I will, and then for the reduced ones where you treat them with DTT, so you essentially get, there's a cleavage that happens, you get to see the light chain and the heavy chain, which is the 23.45 kilodaltons, the light chain, and the heavy one is a 51 kilodaltons. So you get to see this kind of in-depth information when you use high resolution, advanced mass spectrometry. So I'll pause. I think, I've slightly exceeded my time. So that's it I have for today. Thank you for your attention.
Yeah, thank you, Dr. Ravi, for your excellent talk on introducing about the regular role of the CRO in this biosimilar development.